Hello, I'm Stan Finkelstein. I'm pleased to join 4 other members of our research team to offer a short presentation of the highlights of our research that has been generously supported by J-Clinic. Our project is really about finding new uses for drugs that are older and have been on the market for other indications for quite a while. Literally thousands of drugs are currently on the market for specific indications, and they already have had their safety profiles thoroughly studied. And now we have an opportunity to systematically examine very large data sets and apply modern quantitative methods to attempt to find signals of benefit for unmet medical disorders. Our approach is to identify the unmet medical needs that we're interested to serve as targets for repurposed drugs, including Alzheimer's disease, cancer, and COVID-19. To use retrospective observational data from electronic health records and insurance claims to emulate randomized clinical trials. We can think of doing this in 2 steps. First, screening to develop a list of drug candidates of possible interest. And then we will use modern analytic methods to create and balance cohorts and compare these cohorts according to specified outcomes.
Hello, it's Roy Welch, and I'm part of the team, as you can see from the earlier slides. I have 2 topics to discuss concerning our research behind some of the issues related to drug repurposing. We have data issues and modeling issues. So the data issues are here on this slide. The problem we have with data in general, and this type of data in particular, is errors and outlying values. I tend to call that worrying about robustness of the data. We've had examples where there are definitely outliers. And if you, with high dimensional data, potentially thousands of confounders and comorbidities and so forth, each column, each confounder and so on is likely to have some outlying values. And if you then say, well, there's an outlying value, I'll just remove that patient, you end up removing all your patients essentially. So there needs to be some better way to address that, and I think we need to do research on that, and there are methods to consider to do that, to develop a way to deal with this robustness issue. Now, missing data has some similar flavor to it in the sense that with high dimensional data, you potentially have missing data in every feature, every comorbidity, every confounder. And if you say, well, I'll just remove the patient that has missing data, you end up removing all your patients. So we need to address this issue. Imputation is one way, and there are others that are being developed. We need to worry about this situation, and we have lots of missing data. We have bias potentially arising because of the choice of data sets. So we choose a data set in some part of the world or some part of the US, different demographic factors, different ways of collecting the data and so on can produce bias. We need to understand that. We need to work carefully. We need to use validation to figure that out as well. How can we combine data sets in a useful way? When we have data like this, the problem is that if we want to combine it, of course, they're maybe from different locations, but when you're repurposing drugs, you need to have a group of folks who haven't taken the drug and a group of people who have. And the people who have taken it, they've probably taken it in connection with some other health issue. And once you kind of screen it down, finding those people can be very small. So you think you have a lot of data, but when you actually try to repurpose a drug, so since you need people who took the drug and people who didn't take the drug to emulate a clinical trial, it can be quite difficult. And combining data sets with something like meta-analysis or there are other techniques will help, I hope. And then emulation of randomized clinical trials, we have to address the fact that patients come and go varying time periods, they drop out, they come back and so forth. Usually not the case in actual RCTs where it's tightly controlled. So we're looking at tensors to put another dimension in, and various machine learning procedures like recurrent neural nets and LTSM. So the modeling concerns, we, as I said, have many possible confounders and comorbidities, and as we go along, there's the doctor's text notes, images, wearable device, data streams, and so on. So if we high dimensional data, we have that issue arise more and more. How do we select the important variables? Where penalty methods may do it, they create certain bias problems. How do we balance expert opinion with what the data is trying to tell us? So we have the data, it tries to say, these are the important things. The doctors and medical professionals say some other things are important. We may combine them, but this leads to some issues we have to address. The false discovery rate is really, we're looking at the data. We are, if we, particularly with variable selection procedures and so forth, or even expert opinion, experts have looked at data. We are looking at a lot of different hypotheses. This is called multiple comparisons. There are a number of new methods that we're investigating and trying to develop further for our situation. Debiasing methods and knockoff methods, where one tries to understand when to stop doing variable selection and so forth. Accuracy, I don't think a single number is enough. We need to have uncertainty intervals around our accuracy. If we say something 77% efficient or useful, or however we want to phrase it, then it's plus or minus something. We need to understand how to get that number and understand that it's a conservative number. We should be conservative, I think. Bootstrapping is a way, it's computationally expensive and has issues of correctness in certain situations. Then as we use machine learning methods and more complicated methods to find out when a variable is important, to find the variables and their importance, how do we do it? In fairly straightforward regression models and so on, you always have p-values and confidence intervals and so on associated with those models. Much more difficult situation with these more complex machine learning models. So we need to worry about that, because we need to use it to help explain to people what's driving our model and what is important. And finally, transparency and validation. We have to have models that have as much transparency as we can possibly get, and are validated on other data sets and different situations. So doing that, I think, is very difficult. And with advanced machine learning models, transparency is definitely a difficult problem. Thank you.
I am Aamna Shehhi. I'm going to talk about our study, which aimed to investigate the differential effect of metformin in comparison with sulfonylurea on the mortality risk of patients with different coexisting clinical trajectories. Using CBRD, we extracted approximately 200,000 registered patients under drugs of interest. We divided our population into 6 cohorts based on a pre-existing comorbidity, such as COPD, cancer, and chronic liver disease. We also inquired patient baseline information, for example, BMI, depression, smoking status. Our findings indicate that metformin users had significantly lower mortality rates for all cohorts. The beneficial effect of metformin to reduce mortality is higher in the 5-year follow-ups for cancer and chronic liver disease patients.
My name is Shenbo, and I'm going to introduce self-controlled cohort study for high-throughput drug screening. Our interest for self-controlled cohort study is incidence rate ratio, where incidence rate is defined as the number of dementia diagnoses over exposure time. As illustrated in the figure, the inclusion criteria is patients with at least 1 prescription. The treatment group is defined as continuous drug prescription within a 30-day period, and the control group is defined as the same length period before initial drug prescription. In this project, we compared dementia incidence for around 2,500 drug ingredients. Top results ranked by higher 95% confidence interval of incidence rate ratio are presented here. Though dementia drugs appear on the top since patients do not prescribe these drugs before dementia, these results are not reliable since dementia interacts with these drugs. Results from non-dementia drugs reveal association with a reduction in dementia incidence. These drugs can be potential candidates for both observational studies and randomized controlled trials.
My name is Raluca Cobzaru, and I'm a PhD student in the Operations Research Department. When building our model, we are interested in the effect estimation bias due to variables which have some form of association with the treatment outcome or both. Since we're dealing with hundreds or thousands of variables, it would be easiest for us if we could feed all of them into our predictive model and obtain an unbiased effect estimate. However, this is not only time-consuming due to the size of the problem, but it would be very difficult to obtain the desired estimate without removing the variables that would induce certain biases. Choosing which variables to remove can be a complicated process, since correlations between variables need to be supported by medical expertise or some other empirical measure of association. As such, to make our process more efficient, we would like to understand exactly how much bias a certain distribution of covariate correlations, as well as the strength of such correlations, induces in our effect estimate. This would also help us in the case where, in spite of our efforts, we cannot identify all possible correlations between variables, so the selection process would be imperfect. The way we assess the kind of data trimming we need is through simulations. To test our estimation tool, we use the data generating process from a 2020 paper to obtain realistic data sets and augmented the data with extra covariates correlated with the treatment or outcome. We tried different proportions of variable types and strengths of association to inform our future variable selection. And we see, for example, that for a specific ensemble of learners in specific variable distributions, we obtain a bias of about the order of the true effect, even for a small number of strongly correlated variables. This draws our attention to the importance of identifying and removing such variables from our data.
That's Roy Welch again. I want to take a final moment here to gratefully acknowledge the support of the J-Clinic team, both financial support and the staff of J-Clinic in terms of helping us through this process and sharing their knowledge and wisdom and helping us work, making it easier for us to work on this project of repurposing existing drugs, which has the problems you've seen and the hope that you've seen in these slides. Thank you very much.